Hi everyone, this is Eric Appleton. I'd like to welcome you all to our webinar on the TASC Science Item Specifications. So while we're starting, we still have a few minutes. It's not too late. Please introduce yourself through the chat. I see a lot of you have done that, maybe everybody. And I just want to say it's great to have so many people on the call and people from around the state. We have a lot of our teacher leaders on the call, which is really wonderful, and because we're looking forward to getting this information out and getting ideas from everyone on how we can improve science education across the state, and hopefully the item specifications will help us do that. So a little bit of information about the webinar before we jump in. You'll see at the top that we have a hashtag, so hashtag CUNY HSE. If you are tweeting out about the webinar, please use that hashtag, and that'll make sure that we see it and we can respond. So if you have any questions that don't get answered during the webinar, you might, or they come up after the webinar, you could tweet them out, and Mark and I will respond probably, and there's others that might respond as well. So let's go ahead and jump in a little bit here. So just a few notes about the webinar. Everyone except our presenters has been muted, and we're hoping that you'll participate through the chat function with questions and comments throughout the webinar today. If you have a technical question or problem, please send a private chat message to me through the chat function. If that doesn't work, you could email me on the side, and I'll probably get it, but hopefully everything's working for you all. And if you have any questions that you would like us to address during the course of the webinar, please post them in the chat box. Mark is going to be monitoring those questions, and our panelists will respond during the Q&A section of today's session. And then we'll be introducing everybody in a minute here as well. So our goals today are, we hope that teachers and other participants on the call today will leave with a better understanding of the TASC item specifications documents in general, and an understanding of the specific content emphasized in LS14, which is the content standard that includes cellular division. That's the content standard we're going to take a close look at today, and the one that we sent a few chapters in additional reading to everyone who's participating. Our final goals for the webinar is to start generating ideas for how to use the item specification documents and other resources to teach this content on the TASC science test. And so we're looking forward to your contributions and creativity on how to use these documents to improve our teaching. So I'd like to introduce today's presenters. My name is Eric Appleton. I'm a math and science professional developer at CUNY's Adult Literacy and HSE Program. And let me say a little bit about my background here. Before I came to CUNY, I worked in an education program for formerly incarcerated people, and I worked there for 14 years. I've taught all subjects in adult basic ed and high school equivalency, and have been focusing on science and math recently. I've been in the field since 1999. And I'll be hosting the webinar, so if you have any technical issues, send me a private message, as I said. Deedra Arvin from DRC CTB is joining us. Deedra is a test development director. She oversees the development of assessment programs, including the TABE test and the TASC high school equivalency exam, the TASC. Prior to her work in assessment, she taught high school chemistry, physics, and physical science. She has worked in the field of assessment since 2004. Deedra will be giving an overview of the TASC blueprints and item specifications, and will answer questions related to the TASC exam. Did you want to say hi so everyone knows your voice?
Hi, everybody.
And we're also joined by my colleague, Kate Brandt. Kate is an ELA and Social Studies professional developer for CUNY's Adult Literacy and HSE Program. Kate has led numerous curricula planning groups and teaching institutes focused on content-based instruction over the years. She's been teaching and doing professional development in adult education since 1990. Kate will be interviewing our guest presenters about implications for our teaching. Kate, you want to say hi to everybody?
Hello, everybody.
And we're also joined by Tim Jones from the New York State Education Department. Tim is an associate in Instructional Services, and for the last 3 years he has served as the Science and Math Content Specialist for NYSED's High School Equivalency Unit. Tim is certified to teach physics, general science, and mathematics. He has taught high school math and science in both regular education and special education settings, including teaching HSE preparation in a correctional facility. Tim will be giving us an introduction to a specific life science standard on cell division and differentiation. Tim, you want to say hi?
Oh, hi, everybody.
Just want to give everyone a chance to say hi so you know their voice when they come around. And finally, my colleague Mark Trushkowsky is a math professional developer for CUNY's Adult Literacy and HSE Program. Before he came to CUNY Central, Mark taught beginning reading and HSE level math at LaGuardia Community College. He's taught all subjects in adult basic education and high school equivalency and has been working in the field since 2001. Mark, I'm going to ask you to try that one more time. I think you might be muted. Is that possible?
That is very possible. Good afternoon, everybody. I said that this is my first time being on this end of a webinar, and so I appreciate everybody being part of this. Thank you.
So just a quick look at our agenda here. I'm going to do a quick launch to get us into the content that we're looking at for today in the specific standard of LS14. And then I'm going to turn it over to Kate and Tim to discuss evidence statements for LS14, and there'll be a Q&A following their conversation. This will be followed by an overview of the TASC blueprints and item specifications with Deedra, also followed by a Q&A. And then finally, we'll have feedback and maybe a short conversation about next steps. So we're starting kind of with a specific granular look at the item specifications with some specific content around cellular division, and then we're going to zoom out slowly to look at the rest of the science item specifications. So that our goal is to understand how these item specifications work overall and how they might be useful. So we're going to start with a bit of a launch, and this is maybe something similar to what we might do with students if we were jumping into some new content. So the first thing we're going to do here is look at a prediction guide. And so a prediction guide, the way we use them in class is to activate background knowledge, find out what students know, and generate questions about the content that we're going to be studying. So we're going to look at one together. And so for a moment here, you guys are students, and this is something we like to do often when we do professional development, is put ourselves in the position of students and then reflect on what that was like as teachers to think about instruction. So take a look at each of these statements and decide whether you think each one is true or false. You might want to use pencil paper and jot down your answers. As soon as you had a chance to read them and decide whether they're true and false, start entering in the chat box your thoughts. You can go ahead and put your answers in there, comments, questions. So we're going to be quiet for a few minutes to let you read and respond. So by now, I think you've had enough time to read each of these statements. Take a second to write in the chat box whether you think each one is true or false. Like, so for example, Will, I noticed that you put 1, but I didn't know whether you meant that that was true or whether it was false. So for each statement, let us know whether you think they're true. Like for example, Kathy just wrote, I think 1, 2, and 3 are true, 4 and 5 are false. So do you all agree with that? Let us know if you agree and maybe why, or if you disagree, let us know why you disagree. So Sherry and Larry responded and said, well, number two must be false because I think single-cell organisms exist. So that's interesting. And Megan responded that 4 is false because there are different kinds of animal cells. Megan says that 5 and 2 are also false. Maggie says only 1 and 3 are true. But 5 might be true if repair counts as growth. That's an interesting idea. Does repair of tissue count as growth? Okay, so I think we've got enough information out there. Please go ahead and continue putting information in the chat box and discussion. There's a lot of interesting conversation happening already. And I'm going to move to another activity for opening up content in the classroom. And you might not use both of these. Maybe you would, maybe you wouldn't. Maybe you would use one of these or the other. So we just did a prediction guide. And we'll, by the way, we'll look at as what we consider the true and false statements at the end. Right? So we're going to leave this question open for a little while, but we'll come back and look at the prediction guide at the end. So the next thing we're going to look at is we're going to look at some images related to the content that we're going to be discussing. So this is another way of opening up new content in the classroom when you haven't studied it before. So take a look at these 2 images. I'm going to ask you just to look at them carefully and think about what you notice. What stands out to you? What's interesting about what you're looking at? So what do you notice? Take a moment to write in the chat box. What do you notice about these two images? Start writing down the things that you're noticing. So some of the things that people are noticing are interesting. We've got different colors, different shapes. Some have a visible nucleus, some do not. On the right, I see some cell division taking place. That's an interesting idea. Do others see cell division taking place in the right image? A cell with a double nucleus. Wow, that's interesting. I don't know that I see that. I'd be interested for someone to point that out to me. Square and rectangular shapes, around circles on the right. So now I'm going to ask you to start thinking about what you wonder, what questions occur to you as you look at these images. So start writing some questions. Judith got ahead a little bit and asked a question. So are those cell walls in number 2 in the second question? It's a great question, right? What other questions do you have? So here's some other interesting questions that popped up. Is the second picture a closer view or a higher magnification of that first picture? Are they both from the same tissue? How do each of these pictures differ? And here's another question about the magnification. That's an interesting question. Are these at the same magnification or are they at different magnifications? Okay, now consider this image. What do you notice? What questions do you have about this image? And here's one more. Okay, here's a little bit more information. Take a look at this now. What do you notice? What questions do you have? So while you're working on that, I want to talk just a little bit for a minute about why we use images to introduce content. Just a few ideas to think about. Maybe you've already experienced it, but this is a way for, we believe it's a way for everyone to participate because everyone starts in the same place, even if you don't necessarily have particular background knowledge about the content. And everyone, pretty much everyone can look at an image and say what they notice, right, or what questions they have. So it's a sort of a leveling. It has a leveling effect of allowing everyone to participate. The other thing is that everyone notices different things, so we all contribute in our own ways, and we benefit from each other's observations and knowledge. And we already see that happening through the chat box now. We're looking at evidence from the real world, so it's not necessarily, we can think of it as not just textbook information, but this is stuff from the real world. I mean, that's one of the wonderful benefits of using science in adult education. I guess the last thing is that it can take people by surprise. It can promote questioning, and it's just fun to be posed with a mystery. So considering that mystery, what is this? What are these pictures we're looking at? There are some guesses. Maggie guessed that picture A was blood cells. I think there was another guess earlier that those might be blood cells. I'm curious, if you take a second, what do you think the rest of these images are? I give you 5 seconds to make your decisions. Okay, here they are. So the first image is a picture of blood cells. The second is heart muscle. The third is onion cells. And the fourth is tomato skin cells. Wondering if anyone was surprised by these identifications. Okay, I want to thank everyone for your participation in this section of the webinar, and please keep it up with comments and questions related to what we're looking at. I'm going to pass this over to Tim and to Kate, and Tim and Kate are going to be discussing some of the content on the life science test related to the topic of cell division. We'll be having a short Q&A after the presentation, so please post questions as they occur. Mark will be looking over the chat and be pulling some of those good questions for the presenters. So before I turn it over to Tim and Kate, take a moment to look through the following statements. How do each of these statements connect to the images we just looked at? Okay, I'm going to go ahead and pass it over to Kate and Tim. Thank you all.
Okay, so we wanted to give you just a minute to look at the statements again. And what we're going to do here is Tim really has a lot of science expertise. A lot of the rest of us are kind of generalists. So the idea here is that I'm going to ask some questions of Tim about these statements and about the actual content knowledge. So Tim, I'm going to start out with a little bit of vocabulary. I see this word organism sometimes. I see living thing. Can you just kind of define what an organism is?
Well, I can try. An organism is any individual living thing. It could be a plant, it could be an animal, it could be something very large like an elephant, it could be a human being. But it's an individual living thing is an organism.
Okay, that's helpful. And for the very first standard, I see this term cell theory. Could you talk a little bit about what that is?
Sure. Cell theory is one of the most basic theories behind biology. It's really been only understood by people for about 150 years. It has three parts, generally. All living things are made up of cells. We're not aware of any life form on Earth or elsewhere that isn't made of cells. The second tenet of cell theory is that the cell is a basic unit of life for both structure and function. So anything that is a living thing is made out of cells to give it its structure, and all the cells do all the life activities that make something be alive. The third tenet of cell theory is that cells arise from pre-existing cells. So all cells come about from a previous cell splitting into two and growing into two new cells.
Okay, thanks. Could you explain life activities in that second standard? What's meant by that exactly?
Life activities are things are like taking nutrition. All living things need to have some energy to just keep the processes of life going. Taking in oxygen, a way to get rid of waste, and then they're also to really be a living thing in the long run, things need to be able to replicate. So if there's no way to reproduce, eventually a living thing will no longer go on through history.
Okay. So all cells breathe? How do they do that?
Well, all cells need a way to take in oxygen and get out waste, which for all cells, carbon dioxide is one example of waste. For multicellular organisms like human beings, we have all of our cells, the trillions and trillions of cells in our body, are all connected to a source of oxygen through our vascular system. Little capillaries are tiny little blood vessels that have branched off from larger blood vessels, and they actually come in contact with each cell in our body and bring oxygen to the cell and take away waste and bring nutrition and so forth.
But cells don't have lungs, so how do they process, I'm assuming they don't have lungs, how do they process oxygen?
They don't have lungs. One of the important ideas for the TASC test in particular is the connection between cellular respiration, where all cells, whether they're plants or animals, get the energy for life by taking basically sugar and breaking that sugar down into carbon dioxide and water. And the energy that's been stored in the bonds of the sugar is transformed into a usable form by the cell. That's cellular respiration. The other half of that balanced cycle in nature is photosynthesis. Photosynthesis is the hand in glove part to cellular respiration. It's the way that sugar is made by plants taking energy from the sun, takes carbon dioxide and water, and combines those two together and forms sugar, which is just simple carbon, hydrogen, and oxygen. And again, cellular respiration breaks it back into carbon dioxide and water again. Around and around the cycle goes.
Okay. So, and Mark says, so the carbon dioxide I breathe out is the waste from each and every one of my cells?
Yeah, that's exactly right.
Okay.
Carbon dioxide's a poison. If we don't get it out of our system, it'll poison our cells and all of us.
Okay. It's not on here, but can I just ask, do cells have smaller parts inside them?
Yeah, they sure do. Cells are living things, and they have specialized parts called organelles. Now, organelle means a small organ, and it really is named for the obvious analogy to organ systems in our bodies. We have whole systems like the nervous system and the circulatory system and the digestive system, which are made out of different kinds of cells forming different tissues, and then those tissues forming organs, which each have a specialized job to help us as organisms stay alive. Well, within every cell, there are these organelles. You see some of them on the screen now in the schematic diagram below. These are very simplified schematic diagrams, but they are showing the nucleus, the cell membrane, some little pink dots that represent mitochondria, vacuoles, and for the plant cell, a chloroplast. And these are all just a small sampling of the different organelles that are found in each cell.
So can I just ask, do students need to know these organelles and what they do?
Well, there's a place we can see, I think, later on in our item specifications where it's spelled out which organelles students need to be familiar with and what level of knowledge they need to have about those organelles. Deep understanding about how all this works is not the real expectation. Kind of a exposure level of knowledge is what we're really looking for here. Need to know what the mitochondria do, that they're always known as the powerhouse of the cell. That's where the energy is transformed into a usable form, no matter whether it's a plant cell or an animal cell. When we look at the item specs, we can see specifically which organelles are fair game for testing on the TASC test without having to have any kind of definition or anything.
Okay, so that analogy you made between the cell and the organelles and how it's similar to our whole bodies and our body systems, that's really helpful to me, and I would think it would be helpful for students too to think about it like that.
Yeah. not all organisms are multicellular. Once the microscope was invented, the world of pond water became fascinating to many scientists. If you look at a droplet of pond water, you'll see all kinds of living things swimming around that are way too small to see with the naked eye. But it's a whole menagerie of living things that are very small, only one cell in size. So for those organisms, the organelles kind of are their body systems.
Right. I think a lot of students would be really surprised to know that there even could be an organism that was a single cell. And I see here a question from Mark, are single-celled organisms specialized? Can you talk a little bit about what that means, specialized?
Well, specialized, when we talk about specialized in cell theory, we're really talking about in multicellular organisms, cells being differentiated. Not every cell is the same in a human body. We have skin cells and we have muscle cells and neurons or nerve cells, and these cells have differentiated as part of how the DNA codes to replicate more and more cells. As we start out, we're just a single egg, and then that egg splits through mitosis, reproduces several times until we get up to 100 or 200 cells. They're all basically the same. But then at that point, triggers go on in the DNA coding and tell different genes to start producing their proteins and guide the development of different types of cells. So that's differentiation, and that's how we get specialized cells that are responsible for specialized jobs. Now, a single-celled organism, saying is that specialized? Well, an organism has to stand alone. It's going to be its own little living creature, and so you wouldn't really think about it being specialized in this cellular context. It might have a specialized niche in nature that it fulfills. It might, survive best in certain conditions and certain nutrients and so forth. So that word specialized can mean more than one thing, I guess, in biology.
Okay, so from what you were saying, I'm going to ask Eric to go to the next slide because I think you were starting to segue into the second set of standards that we wanted to look at. For these standards, we were thinking to start with this question, how do we go from one cell to a baby? You were just talking about that. I think, our students are adults, so they know about the birds and the bees, but they probably never thought about how do we go from one cell to being such a complex organism. So, Eric, is there a diagram that goes along with this? There it is. So Eric chose this diagram to help us kind of think about this whole question. And I see some things that I would want you to talk about later a little bit more. Fertilized egg with DNA from both parents, and then embryo cells with copies of inherited DNA. Okay, so there's an egg. The sperm and egg come together. There's a fertilized egg. The cells divide. They all have the same DNA. And here we have a baby with all these different cells in it, specialized cells in it, presumably. Eric, can you show the statements that go along with this? The sort of evidence statements? So I'm going to ask everybody to just think for a minute about the picture that we just looked at and then read through these and think about the connections between the diagram and these statements. And feel free to keep typing questions into Mark in the chat. Okay, so Tim, I think I'm just going to again start out with a question about vocabulary. Can you say what a zygote is?
Yeah, a zygote is the name for a fertilized egg. Fertilization is the process where a sperm and an egg come together. Some things we should know and make sure that our students are familiar with are that sperm and an egg are produced by a separate cellular division process known as meiosis, and they only have one copy of each of the chromosomes. Our chromosomes for all organisms are generally in pairs with matching chromosomes with similar genes on each one, but one chromosome generally comes from the father and is going to be carried by a sperm cell, and one chromosome is from the mother, and that's going to be found in the egg. The sperm and the egg magically come together and fuse into one cell, and the chromosomes from mom and dad pair back up and create a new organism with some of the characteristics of each. So the word zygote is a word that we should know, and it means fertilized egg.
Okay, Eric, I'm going to ask you to show us a diagram because when you gave your answer, a question that really comes up to me is I keep hearing chromosome, DNA, gene, and I'm a little confused about each of those, exactly what each of those things mean. I know they're not the other diagram, Eric. It's like it's a little confusing because they seem to be used interchangeably. So I'm wondering if you could explain the differences and then the similarities between them. Sure,
I can do my best anyway. DNA is the material of the basics of life. It is the stuff itself. It is a long, long molecule. It's basically put together from little building blocks. They're called, nucleic bases, or just bases is all we need to know about them for a task. Nucleotides is another word that's used to describe these. And they form long, long chains. And these chains are put together in such a way that they just naturally, due to the electrical charges on the different atoms and the way they've combined, they'll form this twisted ladder double helix that we've come to associate with DNA. So DNA is the stuff itself. When DNA is in the cell between its replications, when it's the cells, it's in the interphase. But during that phase, the DNA is just like a long string, or several long strings that are just uncoiled. But it's unwieldy. How could we, separate into two new cells if we just had these massively huge, long, thin threads to try and separate? So the DNA itself has codes to produce proteins that cause the DNA to wind and twist and then bundle up into special bundles that are called chromosomes. So chromosomes are bundles of DNA. Now, along the stretch of DNA, DNA is amazing, magical stuff that we really didn't only understand now for about 60 years if humans understood how DNA works. So it's really incredible how much we know about it now in a relatively short time. But DNA replicates itself. It can make new copies of DNA, and also has the instructions for how to make all the different kinds of cells that are necessary to produce an organism like a human or a tree. And it also it has all the instructions exactly how to make proteins. Now, proteins are similarly put together out of building blocks. In the case of proteins, there's 20 amino acids, and there's an amazing sequence of how the code for DNA with 4-letter alphabet translates into basically a 20-letter alphabet to build specific proteins. So they're real long chains. The proteins then fold up into very special shapes that'll have electrical properties in certain areas, again, just based on how the molecules are attached to each other and how the electrons end up being bunched together and so forth. And so you get active sites that give these proteins all their special abilities. So that's what a gene is. A gene is a section of DNA that codes for a specific set of amino acids to be put together in a specific order that then will go off, fold up, and become a usable protein for our body. All this DNA is housed in the part of the cell called the nucleus. So I think we covered those 4 terms.
Well, so since you went into proteins, let me just say I'm a little confused because I think of protein as like, milk and eggs.
Mm-hmm.
So why are proteins so important in this DNA process?
Really, proteins are the answer to what DNA does. DNA makes proteins. It has the code for every kind of protein that our body needs. It has codes that tell it, it's an amazing system. There's switches that switch on genes and off genes. Proteins are involved with not just the structure of our body. What we are is mostly protein as far as, we're the cells themselves. All the structures are made out of different proteins. And the proteins also are things like enzymes and have chemical properties on top of the structural properties. So it's an amazing thing. And the DNA puts the proteins together in a way that's much more complicated than we need high school equivalency students to know. But they should know that is the main function of DNA, is that's where it codes for these proteins.
Okay. I'm hearing a little bit of an analogy of the way elements combine to form different compounds. Does that seem like it might be a helpful way for students to?
It is a very helpful way to think of it. Yeah. And
okay.
when you mentioned milk and other foods that are known as proteins, one of the important things to realize here is for the DNA to construct our proteins, they have to have those building blocks. They have to have the amino acids in place and then put those together. Well, our body can make a few kinds of those 20 different amino acids, but mostly we have to take those in through diet. So when you feel yourself craving something with protein in it, maybe some nuts or some beef jerky or something that's a high protein food, it's a signal from your body saying that we need some more amino acids here so we can build some more proteins.
Right. So that's helpful, and I would think that a diagram like this would be helpful to students too, to kind of visualize how all this gets put together. Eric, I'm going to ask you to go back to the statement page to clarify a few other things. Okay. So I guess that I'm wondering if you could talk about number 2, development is a highly regulated process involving mitosis and differentiation. So in that diagram we looked at what points are we talking about mitosis and at what points are we talking about differentiation?
Well, the diagram with there we are. Okay. Well, again, to produce the sperm cell and the egg cell that this diagram starts with, there's a separate cellular division process of meiosis. Then there's the fertilization process, which produces that fertilized egg, and that's called
zygote. Yeah. Can I just ask you to go back for one moment and say,
sure.
So that meiosis has happened. Does every cell in the body have the same DNA except these cells that are produced by meiosis?
That's exactly right. Every cell, whether it's a brain cell or a muscle cell, they all have the exact same set of chromosomes that traces back to when we started out as a fertilized egg.
Okay.
So what you see here, this fertilized egg is developed into a ball of cells. It's an embryo at this point. And at this stage, they're not differentiated still. These are just basic cells that are very similar to the way the egg started out. At a certain point, though, once there's enough of these cells, they start to interact in a way and signals are sent that start turning on different genes in different cells, and that's when the differentiation process starts. And that's an important word for TASC students, is differentiation. And that means that different types of cells are produced through the process of mitosis. So differentiation continues. There's a heck of a lot of differentiation that goes between the ball of cells we see and the baby. They're going to start by making some nerve cells, and then cells of the gut are formed, and it's a very regulated process that is very similar in all animals. It's cell theory traces also to one of the other theories that's basic to all of biology, is that's evolution. And one of the great evidences of evolution is the similarity in how this development happens for all animals across the board. So differentiation is an important idea.
Right. Okay. So that's really helpful to know, because I learned science from a textbook. I don't have that background. And when I saw these diagrams of an animal cell, I was like, wait a minute, are all cells alike? So now I feel like I'm understanding a little bit this process. I just want to review it really quickly. So before this diagram starts, we would have the process of meiosis, right, to produce the sperm and the egg, right? And then they come together, and then there's a full set of DNA, and then it keeps going through mitosis, and then between the ball of cells and the baby, there's this process of differentiation that happens. Is that right?
Absolutely true. That's all those cells. Yeah, every bit of that DNA is not going to be used for every cell. Certain genes are going to trigger the cell to become a skin cell versus a nerve cell, for instance, and they don't even look the same. These cells can look very different. We saw some pictures of muscle cells and blood cells earlier today, and they're very different from each other. But the DNA is identical. Okay.
And so this is an example of sexual reproduction, right? And there's also such a thing as asexual reproduction?
Mm-hmm. And asexual, when you put A in front of a word, it means not. Right. So asexual would be reproduction without fertilization. So asexual reproduction is generally how all single-celled organisms reproduce. They just reproduce through mitosis. They split in half, and part of mitosis, it should be remembered, is that the DNA itself replicates, and you end up with two complete sets of chromosomes. So that when the cell separates into two cells, then each new cell will have a nucleus with a full set of chromosomes, a full set of DNA. So asexual results in cells that are absolutely identical to the mother, and sexual reproduction does not. That can happen in some organisms that are a little more complicated, things like the hydra or something, the yeast. But generally, it's real simple things are going to reproduce asexually. Okay. Although, I guess, Tim, but there's always. I was just thinking of vegetative propagation. Somebody's probably going to say is a way that you can just clip a piece of a plant and grow a new plant from it. Well, that would be asexual reproduction as well. Right. So. Okay.
All right. So I have been hogging the mic a little bit here, and I'm going to let Mark take over now and do Q&A, bringing up questions that people have been putting into the chat. So thank you very much, Tim.
Great questions, Kate. Thank you.
Thanks, Kate. So before, just to clarify, some of these questions are questions that teachers have about their own knowledge, but also some of them are questions that teachers have some ideas about, but they're not exactly sure how to explain it to students. So kind of, Tim, as you hear my questions, maybe think about both of those lenses. So I want to start off with, atoms are the smallest thing, and then cells are the basic unit of life, and proteins are the building blocks. So I think individually I kind of understand those three comments, but can you just talk about them all together in their relationship with each other?
Yeah. The atoms, first of all, are the smallest bit of stuff that can exist on its own. An atom is just put together with the smallest little subatomic particles of protons and neutrons and electrons. They're very, very tiny. As tiny as a cell is, an atom is just amazingly much smaller. So atoms are the building blocks for all materials, for all stuff, including living materials and non-living materials. Any atom is always going to have the same, if it's going to be of a certain element, and all the atoms in an element have the same number of protons, basically. So you can combine more than one atom and get a molecule, another real important idea. And a molecule, for example, would be a molecule of water, which is H2O, 2 hydrogens and 1 oxygen, and it doesn't have any of the properties of hydrogen or oxygen separately. The water molecules make a completely different stuff. So cells are the building blocks of all living things. Now, they're much more complicated than just an atom. They have specialized parts. They have to be able to carry on life activities. They have to have a way to reproduce. And so that's all controlled through DNA, it turns out. So what was the other term you wanted to talk about?
Proteins are the building blocks.
And then proteins, yes. Proteins are not themselves living things. They can't reproduce themselves or anything else. They're just a very complicated molecule that folds up into a specific shape and can do specific jobs. And those are the building blocks that the DNA codes. There's something like 12,000 genes is the latest count. They've actually decided there's fewer genes than we thought there were at one point, but there's still several thousand of them that all code for different proteins that we need to build our cells and to carry on our life activities.
Okay, thanks. So I'm actually going to throw 3 questions at you at the same time because I think they fit together really well. The first one is from the perspective of students, why do I need to know this? And the second 2 are, what are stem cells in the context of what we're talking about? And what differentiates between benign and malignant cancer cells? And the reason why I wanted to put those all together, because I think certainly cancer and stem cells are ways in which students hear about some of these ideas. So I think those 2 questions kind of a little bit get at how to present the first, at least, why this stuff is important. Right. So could you talk about those 2?
Sure. Well, stem cells, first of all, are cells that occur early in the development of a new organism. So at the time of an embryo, they're unspecified cells yet that still can differentiate. So there's a lot of research going on with stem cells. There has been for a couple of decades now, because they're exciting in the idea that perhaps we could find cures for different diseases if we could figure out how to turn on the genes and pick this stem cell and make it produce new muscle cells and new neurons and all the rest of it. So that's stem cell. It's a bit of a controversy because a lot of stem cell research involves using embryo material to get the stem cells. As far as, remind me of the other 2 questions now, Mark.
Well, it's really just one more. What differentiates between benign and malignant cancer cells?
All right. Benign and malignant. Benign means, generally it means good, and malignant means very bad. Benign cancer they're not necessarily cancerous. A benign tumor is one where the cells have been triggered to divide. They're not making the appropriate cells for the part of the body that they're growing in and so forth, but they're not dividing so out of control that they can spread to other parts of the body. So what happens in a benign tumor is the tumor may grow for a while, may stop growing after a while. The body fights off cancer in a variety of ways, and so the body will wrap it in some kind of membrane, so that's why we get a tumor. But those benign tumors are much less serious because they don't spread throughout the body. Whereas malignant cancer is a cancer that the cells will all cancer, the cells are dividing out of control. Some mutation, some change in the DNA code has happened. It can happen through exposure to radiation or the wrong kind of chemical at just the wrong time or wrong concentration, and it causes a mistake in the spelling of those four-letter alphabet that make up our genes. And that causes a cell to just start dividing and dividing and dividing. It's maybe supposed to be a pancreas cell, but it's not making new pancreas tissue. It's making cancer tissue. And if it's a malignant cancer, it can spread to other parts of the body and grow throughout.
Cool. You mentioned, I'm going to try to squeeze two more in. You had mentioned that you talked a little bit about the shape of a cell. And could you talk a little bit about what the shape of a cell has to do, how it connects to the function of the cell, how those two things are related?
Well, that's definitely as cells differentiate. They'll look very different. One great example is neurons. Nerve cells have connections. It's very analogous to wiring in a computer or something. There's axons and dendrites, little branches that grow out of a nerve cell. And actually, the more you focus and think and exercise your nerve cells, the more of these branches you can grow. Has been a recent discovery that's kind of exciting to any educator, I think. I always like to think of myself as actually helping people grow new axons. So the shape of other cells is really just related to what they have to do. Blood cells have that shape. They look like a ball that's had two fists pushed in to push it into the middle, and that allows it to have a lot of surface area. Red blood cells are for carrying oxygen, and that allows it just to do it more efficiently, that shape. So depends on what it is the cell needs to do, and that'll some of it isn't going to be directly that the shape of it will look it just happens to be what its job is, that how it works. Okay.
And so the last question is why this content? Why are we talking about this specifically? Is cells the most important thing, or can you talk a little bit about that choice?
Cell theory is again a real basic concept to help understand biology in general. Biology, the life sciences are now 50% of the TASC blueprint. So when we thought about how to introduce these item specs to folks, we thought, well, since life science is most of it or half the test now, let's pick one area that's pretty basic to it. I also wanted to kind of make the point that when we look at these item specs, that they're not necessarily laid out in instructional arrangement. They're just they're arranged in a certain order that makes sense, but not necessarily in an order that makes sense for teaching them. So seemed like a good place to start.
Excellent. So that's going to be the end of this Q&A. But keep in mind, any questions that we aren't able to answer, because there have been a lot of really great ones, we're going to be writing them up, and wherever the home of this webinar is, we'll have some FAQ, and we'll have some written responses to those questions. So keep them coming. I'm going to turn it over to Eric, but to start that, Eric, I'm going to have you put up the prediction guide. You read my mind. So Eric's actually going to introduce Deedra to talk about the item specs. So take a look at this prediction guide. If you have any questions about any of these, please put them in the chat, and we'll come back to them later. Thanks, Tim.
So hi, everyone. I want to introduce Deedra, but I just want to take a moment just to talk a little bit about why we're focusing on this particular standard. Tim already spoke on that. But just to make it clear, the statements that Tim and Kate were discussing are directly from the item specifications. So those are evidence statements from the item specifications, and they were included in the readings that we sent around. And so one of the things that Deedra will be doing is explaining how item writers use those evidence statements and the other parts of the item specifications to put the task together. And so please, while Deidre is talking, please continue to put questions in the chat and consider the goals that we have here, which is about understanding these item specifications and how they might be useful for us going forward in improving our science education. So go ahead, Deidre. Thank you.
Hi, thank you so much. I think we're going to move forward to the next slide for a few questions about the item specifications. So we want to talk about what is covered in the TASC science test. And if you look on page 3 of the item specifications document, the blueprint for both the current forms that are D, E, and F and the upcoming forms that will be out soon, forms G, H, and I, you can see the blueprint, which identifies the domains, which are our reporting categories. So Earth and Space Sciences, Life Sciences, and Physical Sciences, and then some subdomains or groupings of topics within each of those reporting categories, and the percent of the blueprint, the percent of a test form that would be made up of each of those subdomains and domains. So you can see that from DEF forms to GHI forms, we are making a little bit of a shift. So instead of 40% Earth and Space Science, 40% Life Science, and 20% Physical Science, we are shifting as we move to GH&I to 30% Earth and Space Science, 50% Life Science, and 20% Physical Science. And the reason for that is that we looked at, with TASC being a high school equivalency test, it's very important to consider what's going on nationally as far as the expectations of students in high school science. So one of the things that we were noticing when we were doing our analysis of state standards in high school science and state assessment in high school science is that life science really is getting that privileged space in curriculum and in assessment over earth science and physical science. So we wanted to adjust our blueprint to reflect those expectations of curriculum. So we increased the percent of life science to 50%, and we decreased earth and space down to 30%. So earth and space sciences are just a little bit more than physical sciences right now to reflect that those are more equal in distribution to each other rather than to life science in curriculum and assessment nationwide. If we move further into the item specifications, we understand that item specifications are used to define the important characteristics of test questions that are developed for each of the standards. So a test is just a tool that allows us to measure what examinees know, understand, and are able to do relative to the content of the standards. Item specifications help us to clarify the focus of what is to be assessed so that we can make sure that our assessment measures the standards that we intend for them to measure. So item specifications provide information about what items can include and what they cannot include. So we have some assessment limits, which I described some of the content that is not to be included in a test question that assesses a particular standard. If we look at the item specification on page 51, this is the first. There are actually 2 pages for this particular standard, the LS14 that Tim was talking about. We can see that in the item specifications we have the domain and subdomain information as well as the standard, which includes the code and also the descriptor for that particular standard. We also see an emphasis level, which tells us how much focus is included on this particular standard in any given task test form. So if we see an emphasis level of high, that means the pool of items that we draw from to build our task test forms has quite a few items aligned to that particular standard. If the emphasis level is a medium, that means there are not quite as many items for that particular standard, but there are several in the pool from which we select a task test form. If the emphasis level is low, that means that particular standard has fewer items in the pool, not very many, just a few items in the pool that is used to select a task test form. So if you are looking at a low emphasis level standard, you are not likely to see more than one item for that on any given task test form. And there might be some task test forms that don't have any items for a given low emphasis standard. The high emphasis level does indicate that it can be found more heavily within the task test forms. It does not mean that that is the only content that should be instructed for examinees who are preparing for the task test. That just means that there are a lot of more items aligned to that standard in our pool. If you look down to the next box, it has a light gray shading, you see the evidence statements. So a lot of these statements you can see on the slide that Tim was talking about, describing what examinees are supposed to demonstrate understanding of, or what they are supposed to recognize. These evidence statements really give you more information about the standards and what is expected from examinees for each of these standards. When I would be planning instruction using the item specifications, the evidence statements are my go-to place. So I would look at the evidence statements within each given standard and also look across standards to see where those evidence statements overlap and where I might get the most bang for my buck by instructing students on things and using the interconnectedness of the evidence statements across standards in order to instruct students. If you move forward to the next page, page 52, we can see the rest of those evidence statements. We can also see a box for assessment limits or content constraints, and that identifies some of the topics that are beyond the scope of an assessment item measuring this standard. So in LS14, we see that the stages of mitosis and meiosis are beyond scope. So examinees are not going to be asked to describe or identify the stages of mitosis or the stages of meiosis. They are also not going to be asked any information about organelles other than the 6 that are listed in the evidence statement section above. So if you look at that, it says that specialized structures may include cytoplasm, mitochondria, ribosomes, the cell membrane, vacuoles, and the nucleus. Any other organelles are not going to be included. They are beyond the scope of this particular standard. The DOK, that stands for depth of knowledge, and it refers to the cognitive complexity of the test questions that students might see on a task test form. When we look at a depth of knowledge, one, we're thinking about recall, information where students are, they have the information, you either know it or you don't. You're applying a very rote procedure that the examinees will know the information and easily be able to process the information. That would be a depth of knowledge 1. A depth of knowledge 2 is more of a comprehension. So they have to understand information presented within the test question in order to, and use that information in order to answer a question. If you want to learn more about depth of knowledge, the depth of knowledge that we use is based on Norman Webb and his model of cognitive complexity. The next part of the item specifications is the stimulus. And as you can see in this particular standard, a diagram is highly recommended, which means most of the questions that assess LS14 are going to include a diagram that students are going to use in order to answer the question. In science, a scientific calculator is provided. That is the tool that is available to all examinees for all questions in science. The entire science subtest allows a scientific calculator. Examinees may not need it for most of the questions on the TASC test, but it is available for them. The next box you see is a sample item stem. The stem is the part of the test question that asks the question. So if we are providing information that students will use to answer a question, that's considered the stimulus. So it might be a paragraph of information, it might be a table, it could be a diagram, it could be a graph. There are different ways that we can present information that examinees would use in a stimulus, and then after that stimulus you would see the stem, or the question that examinees are asked to respond to. In a multiple choice question, that would then be followed by answer options. So a sample item stem in the item specifications, those are going to just be the questions that examinees might be asked to respond to. In some of the standards for the item specifications, we then follow that sample item stem with also some sample questions. So if we move forward in the item specification, we can see that after LS14, we have LS15, and then we keep going just a little bit more. If we move to LS21, after the sample item stem, you can see a sample item, which provides you with the full test question, including the stimulus information and the answer options, or the way that that would be presented as a test question. For this example, LS21, you happen to have a sample item for an online version of an item, and then that is followed on the next page by the way that an item would be presented in print for that same standard and that same skill that is being presented.
Deedra, can I jump in and ask a question? So would you recommend that teachers use these sample questions with students?
These sample questions can be used with students, but we need to remember that those questions are just samples. So they should not form the foundation of any curriculum that is used, but they can give examinees an idea of the ways that some questions could be worded. However, these particular questions are released items. So they are available publicly, which means they would not ever be found on a test form. So if you teach your examinees only the content found in these sample questions, you would be doing them a disservice.
Okay, thanks. Do you want to talk a little bit about how you would recommend to teachers how to use these specs when they're planning their curriculum?
Yes. I think it's important to look at the evidence statements and see what it is that examinees are expected to demonstrate understanding of. And you can look at those evidence statements across the different standards within a subdomain, because there's likely to be quite a bit of overlap and interconnectedness within a subdomain, so that you can find ways that you can teach topics and be able to cover more than one standard at the same time. Because I know you don't have enough time in your instruction to go through and teach each of these evidence statements separately, nor would that be the best way to help examinees understand each of these topics.
So in other words, it would be good to try to create a course that starts with basic information first and builds up to more complex, and to use the statements as a way to kind of check with yourself, like, is this understanding that students are expected to have? Does that sound right?
Yes. And then you can check back within the assessment limits or the content constraints as just a quick check to make sure that you are not going beyond what is expected in your instruction. You don't want to go so deep in your instruction on a particular topic that you hit that beyond scoped information.
Right, right. And then at the same time, you wouldn't want to leave out things that kind of help students connect the dots, it seems to me.
Right.
Yeah. Okay, so we're going to turn it over to Mark for Q&A. Thank you.
Thanks, Deidre. I'm kind of going to piggyback right on what you and Kate were just discussing. So before the lesson planning happens and using the evidence statements, can you talk a little bit, because the overwhelming question is, if I'm teaching, say, for example, 3 hours a week for 12 weeks, what are the core ideas? What should I focus on? And how can the item specs help answer that question?
So the core ideas are the foundational knowledge that every examinee needs to be ready for college and careers. So you want to make sure that you're touching on that foundational knowledge, and one of the ways to identify some of that is in looking across the evidence statements, looking a little bit at the emphasis level. A lot of the standards that are coded as high emphasis are coded that way because they are foundational information that is needed to build up to some of the other standards. So looking across the item specifications and really, kind of digging in and tearing those apart and looking at the information that really is foundational to build up to other understandings is very important.
And so
Can I jump in?
Oh, yeah, I'm sorry, Tim, go ahead.
Oh, I just wanted to make one point here. The vocabulary of the evidence statements themselves actually is an important thing to consider, because the rules of the game for the tests that we're going by here are, if a word appears in the evidence statements, then that's considered to be fair game for testing, that it wouldn't need definition. So words like mitosis and differentiation and molecule, these are words that wouldn't have to be defined in any kind of reading passage or any kind of diagram to help understand it. It's just the expectation is that the examinee would be familiar with them. So that is an important idea. If you're wondering if it's something I should cover or not, well, if you see it in the evidence statements, then it's fair game.
So to piggyback on that, there was in the chat, Tim, during your part, a question was asked, do the students need to know which bases bond with each other in the DNA code? And I know you both could answer that question just by answering it, which I'd like you to, but also can you say where in the item specs would the answer be? Because I want, because we have this access to you right now, but we're going to be making sense and working with this document beyond that. So, yeah.
Well, I think that particular question was answered in the assessment limits if I saw it up there. But no, even though, yes, right here in beyond scope, the specific pairing of the nucleotides, you don't need to know that A pairs with T. And although I love the apple tree and the garage car,
that was really nice.
Some of the great ideas. And we don't need to know the specific nucleotide names. We don't need to know that it's adenine and guanine and so forth. We don't need to know the definition of translation versus transcription. We do need to know that protein is synthesized, but the details of how that synthesis happens are beyond the scope for high school equivalency. So the stages of meiosis, the stages of mitosis, again, that seen some nice pneumatics. PMAT, that's the way I memorized it back when I had to memorize the prophase and the metaphase and all the rest, but we're not requiring that level of detailed specificity here. We're really looking for more foundational understandings, the ideas that are more connected to other ideas in science, and to at least make this scope, manageable. We can't just say explain the universe and give 2 examples. It's has to be some limits.
Okay, thanks. So Deidre, I wanted to go back a little bit to that idea of say a teacher has 36 hours. I think some ideas that teachers may have about how they can focus is to, for example, not teach physical science because that's only 20% of the questions, or only teach the high emphasis standards because there's a greater probability that those are going to be on the test. Could you speak to those ideas?
Yes, so the idea that you would not teach physical science, as a former physical science teacher, that makes me want to cry. I would encourage you not to skip physical science, not only because it's really super fun to teach and learn, but also because the content that is included in physical science is that foundational knowledge that we believe is important for examinees to be considered college and career ready. So where we do see less emphasis within the blueprint, what is there in the blueprint is that foundational knowledge that is really, truly important. So you don't want to skip over that. You also don't want to just check off and go through and say, I'm going to teach this high emphasis standard and this high emphasis standard and this high emphasis standard, and only teach those standards that are high emphasis, because again, you're doing a disservice to your students in terms of their understanding, because most of these standards are interrelated, and knowing the information from one standard can support an examinee's learning in another standard. So you don't want to focus only on those high emphasis standards, because you can be leaving out important information that will help a student to make those connections that allow them to have deeper understanding of the content they are learning.
So one of the in one of the pre-reading activities that we gave some of the participants before the session, there was some conversation about how challenging it may be to use that with students, and I wanted to particularly talk, so for example, one of the things that was in the reading is the stages of mitosis, and we can see that's beyond scope from the standard. And my question is, what advice do you have for teachers trying to match lessons and resources with the evidence statements? Because there's nothing that's out there that's perfectly aligned, and kind of what advice might you give folks?
Well, Tim has a lot more experience doing that particular matching than I do, so I would love to hear his response to that question. But I think the way that I approach it is in breaking those evidence statements apart and trying to create kind of a concept map based on the evidence statements, and then try to match my units and lesson plans to that concept map rather than just to specific evidence statements individually.
Yeah, I think that's good advice. I think you really do have to kind of start with looking at these evidence statements, and the concept map is a nice way to kind of group them together. Certain ones are related to each other and would connect up well in a lesson. And the other point is the vocabulary would be another thing to stress. Again, if it's mentioned in these evidence statements, it's fair game vocabulary. So just going through and always remember that oftentimes our students have been unsuccessful in the past in these classes, and many times it's because of deficiencies in what we would think of as basic vocabulary. So I would first pick out related concepts, try to get a picture of what the lesson should be framed around, highlight those important vocabulary terms, and then go look at materials and see what's gonna fit it for you. And if you look at Collect Ed New York, hopefully you'll see a growing list of materials that people have found useful.
Great. So my time for questions and answers is over, so I'm going to throw one more question that just has a yes-no answer, which is, are there DOK3 questions on the TASC science section?
Yes, there are some DOK3 questions.
Okay. Thanks. So I'm going to turn it over to Eric. Thank you both for your answers.
Thank you.
So hi everyone. So we're going to start moving towards closing. A couple things before we close. So any questions that that our presenters were not able to answer, we're going to do some work to answer those questions in text, and we'll distribute them all to the group. I want to show you a couple places where you can get resources after today. I also want to let you know that if you did register for the webinar, it wasn't required to connect, but if you registered through Eventbrite, we'll be distributing resources through email. We'll also put up our email addresses here at the end, and so if you didn't get it, please note our email addresses and contact us for resources. So let me switch back to the PowerPoint real quick here. So a few resources that you may or may not know about. So CollectEd, which Tim just mentioned, that address is here. There are a lot of great teaching resources there, and the recording of this webinar along with the PowerPoint will be there, and anything else that comes up as a resource connected to this webinar. And here's a link for the item specifications and the blueprints. Also the Next Generation Science Standards, which a lot of these standards came from originally. There's also this document, Framework for K-12 Science Education, which a lot of us have found useful, which is sort of a prose introduction to the standards. It's connected to the Next Generation Science Standards, but it's written in paragraphs that's organized by topics, and sometimes it's a little bit easier to read as an introduction to different topics, evolution through natural selection, for example. And it gives a nice introduction to some of these science topics. One resource that we've used a lot recently in some of our curriculum at CUNY are the retired regents examinations, which are available here at this address and can be freely used. And they're nice, though they aren't aligned to the TASC. They do have a lot of the similar content and can be useful in instruction, and then we've been using them in our evolution curriculum recently. And then finally, Learn.genetics is a great resource for teaching and learning about introductory genetics, and so we recommend that as well. I want to show you quickly where you can find these resources. Let's see here. I'm going to share. If I remember how to do this, forgive the pause here. Let's see here. Hopefully you're looking right now at the free conference call login screen, I think, which you connected to in order to get on the webinar. If you look here under meeting resources, you'll see some links that I just mentioned. These are all here. And what's also here now is the PowerPoint that we looked at. So it's called the TASC Science Item Specifications webinar. But this PowerPoint, if you just click on download here, you can access that whole PowerPoint. If you have trouble finding it, go ahead and email me. One more thing before we close up here. We wanted to get some feedback from you. So, was this useful? How could it be improved? Should we do more of these webinars on the item specs or other topics on the TASC exam? And feel free to give some feedback in the chat right now. It would be useful. You can do it privately if you like to the host, or that's me, or send it to the whole group. We also will be emailing out a survey, a simple survey on what you thought was useful about the webinar today, what could be improved, and questions you might still have. So please give us some feedback, whatever means works for you. We really do appreciate it. And then finally, we're thinking about next steps. What's useful for us to do as teachers across New York State to improve our instruction for the task? One small idea we have is to start to collect some of these item specifications and think about them maybe across the standards, to bundle them in terms of topics and how put them in order and how we might teach them, and then start thinking about teaching ideas. So we put together a Google document, which you can find through this link here. It's sort of an odd link, but you might jot down this link. I'll come back to it in a moment, but I wanted to show you what the document looks like. let's see if I can get it to come up here. So we've put together a Google Doc that's really open access that has these standards. Just for now, from this particular standard, LS14, and you'll see the particular standards, evidence statements on the left, and then in the middle, some of the framing questions that we might use. And one of those could be, for example, the one that came up in case discussion with Tim, which is how do we go from one cell to a baby, and then other framing questions we can use to teach this content. And then finally on the right side you'll see a bunch of teaching resources. Kate has gotten this started by putting in some ideas of her own, but we're inviting you to participate and add your own ideas into this document. And so there's a lot of great ideas that came up through the chat, and maybe as a final way to capture some of that, you might go to this document here, and I'll give you the address again, https://goo.gl/MHJCU_X, and that'll get you into this Google document. You don't need to log in or anything, and you could just start adding your ideas so we can crowdsource this a little bit. Anyway, our contact information. Okay, so just so you know, this is a work in progress. It is a draft. We've just got started doing this document for teaching ideas. So it will change, and we will be adding some information that's not there yet, and then seeing how we might finalize it and distribute it if it does become useful. Anyway, our contact information is here. Everyone here on the call has offered their email address for follow-up conversations, so please contact any of us with questions or comments. And I want to thank everyone for your participation today.